Hello, welcome to my presentation on bioprinting in tissue regeneration. First, let's talk about the skin tissue. Skin tissue is comprised of many different types of cells, however much of its volume is what's called the extracellular matrix, or the ECM. It's a collection of protein fibers such as collagen, elastin, and others in a proteoglycan gel. This creates a mesh network close to cells. Its structure is secreted mainly by fibroblasts, however chondroblasts and osteoblasts also secrete ECM components. The ECM is a vital structure of tissues as it provides the mechanical support needed for cells to anchor and to take a specific direction and shape. Furthermore, the ECM filters molecules which establish microenvironments for the cells. It can store growth factors which promote or inhibit specific cell behavior. Your skin provides a chemical, biological, and mechanical shield against the outside world. The epidermis is made up of 5 types of cells, most importantly keratinocytes and stem cells. The dermis of your skin contains mostly collagen, but is also comprised of fibers, fibroblasts, and other connective tissue cells. The hypodermis is a combination of fat and connective tissue. Between the two top sections is a thin layer of basement membrane, a layer of ECM. Its main function is to bind the epidermis to the dermis. Deep second-degree and third-degree burns involve the damage and depth of both the epidermis and dermis layers of the skin, destroying its integrity, structure, and functioning. Such deep wounds cannot heal themselves. There are many types of junctions between cells and their environment found in the skin. These are desmosomes, gap junctions, focal adhesions, and hemidesmosomes. Desmosomes are junctions requiring J-shaped proteins called cadherins, proteins that link cytoskeletons of adjacent cells together. Two cadherin molecules hook each other, anchoring the cells together. These adhesions perform a structural role, supporting tissue integrity and allow for even distribution of stress within the cytoskeletons of adjacent cells. It also plays a role in cell signaling and gene expression control for limiting proteins needed for transcription. Gap junctions between the keratinocytes are formed by connexons. These are two black structures made by a ring of 6 transmembrane proteins between two cells. This allows for easy transport of solutes and molecules between cells. However, this also includes toxins and pathogens, so if a cell is dying or infected, neighboring cells are also in danger, and so have to shut down these junctions before being damaged themselves. The binding of the epidermis to the dermis is done through the basement membrane using focal adhesions and hemidesmosomes. Both use integrins, structures containing glycoprotein subunits. These adhere a filament from the cytoskeleton of a cell to the basement membrane. However, they are attached to different filaments. The basal surface of the epidermis has a set of integrins presented that are positioned on the surface of the cell. They bind the cell to the ECM and create a mesh network. Both these junctions serve as mechanical sensors to forces like shear and tension. After a burn, the golden standard for treatment are full-thickness grafts. There are several methods already available for skin grafts. Autografts and allografts are commonly used to provide long-term methods of healing. Methods like xenografts and isografts are also performed, however there are complications found with these types of treatments. A full-thickness graft contains both the dermis and epidermis. After appropriate harvesting, the autograft is cultured and the wound bed is cleaned. Then implantation can begin. As the components of this graft are the patient's own and rich with components like collagen, elastin, etc., the wound bed begins to attempt reconstructing the skin, using the graft as a temporary scaffold. To further aid this process of adhesion, the graft can be treated with a fibrin gel, which aids hemidesmosome adhesion. Sutures and staples stop the graft from moving, a vital feature as tissues require stability in the formation of desmosomes and hemidesmosomes. The pressure of the fixation promotes cell migration, which further helps skin regeneration. These features help support a quicker recovery and reduce the chances of adverse effects. Many alternative methods of skin substitution have been researched and performed, but the most novel is skin bioprinting. Bioprinting is a method of producing a graft combining cells, a biomaterial, and growth factors needed to stimulate the cells left in the wound bed to regenerate the lost tissue. This combination is called a bioink. An image of the patient's skin is made using computer-aided design software and is used to choose the right bioink depending on the skin structure. The bioprinter deposits bioink filaments layer by layer following the skin image. The graft is then cured and reinforced through cross-linking. Many methods and materials of bioprinting exist, with the most research being extrusion-based bioprinting using alginate hydrogels. During extrusion-based bioprinting, the bioink is expelled through a syringe-like nozzle which can be used with a variety of different polymers and cells. The alginate hydrogel structure is similar to the ECM, which provides a physical stimulus to the cell to behave naturally. The hydrogel could be further improved by adding chemicals and polymers to provide the desired graft properties. The use of this biomaterial is very promising, with the ability to incorporate different cells, support cell differentiation, density, and survival within a graft after the printing process. The stimulus given by the graft in its cells urges the cells in the wound bed to release signaling molecules as they would in normal skin. These molecules are called cytokines. The signaling molecules bind to receptors that are specific to them on the surfaces of keratinocytes and fibroblasts, thereby activating them. The activation of the receptors triggers signaling pathways within the cell, usually with the help of secondary signaling molecules. The result of this pathway is a change in cellular activity, which may be migration, proliferation, as well as releasing its own cytokines. Different combinations of cytokines deliver unique cell behavior. The main types of cytokines are interleukins, tumor necrosis factors, growth factors, and interferons. Having live cells in the graft will trigger the cells in the wound bed to migrate into the scaffold and begin tissue regeneration. The signaling molecules can be delivered to target cells through different types of signaling methods, such as paracrine, autocrine, and endocrine. Different methods can be used for the same cytokine, however endocrine signaling is not often used due to its global effect. If the cells of the wound bed do not interact with the graft, the location could become a place of infection and rejection. The immune system would then have to fight off the implant or pathogens. The signaling molecules trigger many cellular behavior, as well as the immune response. This is how the cells of the wound bed communicate with the cells of the immune system. Despite allogeneic closely resembling the ECM structure, the host cells recognize the foreign surface, leading to the activation of the innate immune system, which tries to destroy the non-self material. The response occurs at the blood-material barrier and is called the thromboinflammatory response. It consists of 3 different processes. Right after implantation at the blood-material interface, the body coats the surface of the implant in plasma proteins. This process is called protein adsorption, and the resulting layer is called the protein corona. Immediately after this, platelets adhere to the protein layer. This binding triggers them to release cytokines that aid the immune response by recruiting leukocytes to the site. The released chemicals also cause the blood to clot around the foreign body. This is the first step in creating the thromboinflammatory environment for later escalation. The complement system is described as a cascade of protein interactions, the function of which is the elimination of non-self material from the body. It is part of the innate immune system and is important for urging immune cells to clear the body of foreign cells and structures, promoting inflammation to support this. Complement can be activated via 3 pathways: classical, the lectin pathway, and the one triggered by allogeneic hydrogels, the alternative pathway. They have different methods of activation, but all 3 have the common step of producing C3 convertase that cleaves C3 into C3A and C3B. If the next steps aren't inhibited, the cascade of enzymatic reactions continues, during which byproducts are made. Most byproducts have pro-inflammatory and prothrombic effects. For example, C3A and C5A stimulate leukocytes to release more pro-inflammatory cytokines, which trigger blood coagulation, urge further complement activation, as well as stimulate further platelet adhesion and their release of cytokines. Different parts of the process escalate one another and create the ideal environment for the thromboinflammatory response to initiate. Neutrophils are attracted to the site by the cytokines released by platelets. The neutrophils then recruit leukocytes, who then recruit monocytes found alongside capillaries. Monocytes differentiate into M0 macrophages when they enter the tissue. Further polarization occurs after its interaction with cytokines. Macrophages exist in 2 main types. M1 leads to chronic inflammation, angiogenesis, and inhibits the production of fibers. It is also associated with tissue damage and cytotoxicity. M2 is anti-inflammatory and also promotes angiogenesis. It is also responsible for ECM integration around and inside the material. M2 macrophages are vital for tissue repair. The hybrid type of macrophages is still being researched. It contains varying levels of both, exhibiting a production of isolating ECM and immature angiogenesis. It is associated with biomaterial rejection. The polarity of macrophages is a spectrum, and the cells can exist in multiple states. We will discuss the 2 main types moving forward. The type of cytokine defines what type of macrophage an M0 will differentiate into. Pro-inflammatory cytokines trigger M1 polarization. The structures on the surface of pathogens like lipoproteins, lipopolysaccharides, PAMPs, and DAMPs also trigger this proliferation. Both PAMPs and DAMPs are recognized by transmembrane proteins called pathogen recognition receptors. The main family of PRRs are toll-like receptors. TLRs are expressed by both types of macrophages. The cytokines released by M1 type macrophages are pro-inflammatory. Anti-inflammatory cytokines push type 2 polarization. This can also be induced by the macrophage engulfing apoptotic cells. Furthermore, immune complexes can act alone or together with DAMPs to trigger M2 polarization. M2 type macrophages themselves release anti-inflammatory cytokines. For the ideal integration of biomaterial, the implant should incorporate cytokines that trigger M2 proliferation, as this type is responsible for supporting tissue repair, ECM assembly around and within the material, as well as vascularization. The anti-inflammatory properties of the cytokines aid these processes, leading to better implant acceptance. Testing biomaterials prior to implantation is crucial. The properties of the bioink have to be carefully assessed. We'll talk through a small list of properties needed to be reviewed. The mechanical properties are tested using a rheometer, which allows us to understand the stiffness, strength, viscosity, and other parameters vital to bioprinting. These parameters greatly affect the quality of the final graft. High stiffness stimulates cellular migration and differentiation, helps create junctions and assemble the cytoskeleton. High viscosity helps reduce shear stress on the cells during printing, increasing the cell's survival rate. However, the mechanical properties of the graft must be similar to the natural environment for successful integration. The barrier property of the bioink is tested using fluorescently labeled molecules, where specific molecules are tagged with a fluorescent marker, which then allows us to see how the molecules have traveled through the material. The marker molecules release energy in the form of light. Different molecules give different colors. This test is very sensitive and selective, producing high-quality results. These findings are then compared to native skin models. The barrier property of the graft is vital in allowing the movement of nutrients, factors, and signals. This property can also determine if the graft exhibits the same shield property against irritants as natural skin. MTT assays are performed to measure cell viability. They measure how much of the MTT region has been metabolized by the cells. The yellow region is reduced, producing an insoluble purple solution called formazan. The opaqueness of formazan is directly proportional to the number of live cells. A high number of live cells is vital for wound healing. The biocompatibility of a material is extremely important, as it allows us to know the implant will not harm the body. Alginate hydrogels are clinically proven to be biocompatible and can be further improved by incorporating other chemicals in its structure. To analyze cell behavior, staining tests are done. Examples of staining include H&E, Ki67, and others. One of the most commonly used in dermal evaluations is immunohistochemistry. Similarly to fluorescent marking, a target cell or molecule is stained with a pigment for easy detection. Antibodies are used to detect proteins which can be related to specific examples of cellular activity. These proteins are direct selective markers showing different cellular behaviors. Irritation tests review how the bioprinted skin would interact with natural skin. There are several protocols that describe different testing techniques. The graft or its extracts are applied to human or rabbit skin models and monitored for irritation. This test is vital to check for biocompatibility, and alginate hydrogels are known to be non-irritating. The biodegradability of the alginate hydrogels can be evaluated using another set of protocols, specifically for manometric respirometry. The hydrogel is immersed in a mineral solution with microorganisms within a closed respirometer as a food source. The respirometer is incubated for 28 days, during which the microorganisms metabolize the specimen. The decrease of oxygen is monitored and is used to calculate the degradation index as a percentage. The degradability must be higher than 60% to be considered biodegradable. For alginate hydrogels, the percentage is 61.1. Alginate degrades into non-toxic sugars which can be easily absorbed, so there is no need for surgery. This property also lowers the risk of chronic inflammation and foreign body response. The wound fluid and surrounding neutrophils coat the implant in a protein layer which triggers macrophage to differentiate into foreign body giant cells, which then recruit fibroblasts. They begin to encapsulate the implant, cutting off all vascularization and tissue interaction. 3D printing is a developing field. Some setbacks still exist. The main adverse effects reported following the implantation are rejection, improper function of the skin, and difficulties in maintaining cell viability during printing. During the first week after transplantation, vascularization of the graft allows for immune cells to analyze the graft. If using a cell bank, even though tissue matching is possible, the cells do not have identical MHC proteins on the surface, which appears foreign and harmful to the body. The body begins to send immune cells, such as T and B cells, to eliminate the threat. T killer cells trigger the cells presenting the wrong MHC surface proteins to undergo apoptosis. T helper cells enter the transplant through the blood vessels to stimulate macrophage differentiation, which also causes chronic inflammation. B cells produce antibodies against the foreign antigens. Both B cells and T cells disrupt vascularization of the new tissue, leading to chronic rejection. Autologous grafts are safer, but also pose risks of rejection. The skin is a complex organ made up of many types of cells and structures. Hair follicles and sweat glands are vital for skin functions like UV protection and temperature regulation. Big sections of skin that aren't able to perform these functions may develop conditions such as anhidrosis, infections, sunburns, and many more. Melanocytes, cells producing the pigment melanin, are hard to incorporate in the bioink due to the challenge of ensuring their functioning and viability both during and after the bioprinting process. The lack of cells and structures may make the skin look not normal, which may cause great mental distress to the patient as well as the disrupted functionality. The process of bioprinting exposes the cells to thermal and mechanical stresses. Some nozzles require a higher amount of force than the cell can handle. These triggers can decrease cell viability, which would directly affect the quality of the graft as well as the healing process of the patient. Advances in bioprinting include ways of minimizing rejection, such as improvements in scanning of the patient's tissue, creating better methods of cell culturing, and optimizing the choice of growth factors. New methods of bioprinting help provide the process with ideal physical conditions to maximize cell viability. Researchers are developing methods of incorporating more cells and structures, as well as improving the graft's biomaterial properties for new methods of cross-linking and polymer creation. Bioprinting is a rapidly growing scientific field and is not limited to skin only. It brings us closer to a future of safer, patient-specific treatment for a variety of conditions, fast and effective recovery, which is inclusive and accessible to all. A lot of work is still to be done, but the future is closer than we think. Thank you for listening to my presentation.